In this video lecture, we're going to look at innate defenses. Remember, this is our non-specific immune system where all the cells and chemicals involved are going to behave the same regardless of what the pathogen is. There's no special population of cells to fight a particular pathogen in a different population. That's adaptive immunity. This is more generalized in that sense. So we're looking at a bunch of the actors in these, or the soldiers in this innate defense. So we'll look at phagocytotic cells, mast cells, and natural killer cells. And then we'll also look at all the chemical defenses that we can use in innate defense. So let's first start with the phagocytotic cells. These are the neutrophils, the macrophages, the dendritic cells, which we haven't talked about before, but dendritic cells are related to white blood cells. They came from the same hematopoietic stem cells, but they never entered blood, so they're not considered a white blood cell. And these guys are found typically in tissues that come in contact with the external environment. So places like the skin, the lining of the nose, the lungs, the stomach, the intestine, any place again where you get in contact with the external environment, you'll find dendritic cells. Mast cells are also another one of these cells that came from hematopoietic tissue. Again, they didn't enter blood like the white blood cells, so we don't consider them a white blood cell. But they also function similarly, and they're going to be seen more related to the same function as a basophil. Now, the first thing these phagocytotic cells have to do is they have to find the site of the infection. And think of neutrophils in this case. They're floating around in the blood, doing their own thing. They have no idea what's going on outside the blood. So we got to make sure they find that way. We have to direct them to the site. So the way that happens is a process called margination. This is the clinging of the phagocyte to the inner wall of the capillary. So in other words, they're again floating around here in the blood, and we've got an infection here. And so we need to tell the white blood cell, hey, get out. And so we have these CAMs, which are called cell adhesion molecules, that signal the location of inflammation. So think of it as a road sign. It's like, hey, exit here. This is where the inflammation or the infection is going on. And so they're like little blocks that stick out of the inside of the cell, or excuse me, the endothelial lining. And that way the neutrophil will stick to the endothelial cells. And then once it sticks, then it starts looking for places to exit, and that's the process of exiting is called diapedesis. And we talked about this before, the idea of squeezing themselves between adjacent cells of the endothelial to be able to get out of the blood. And then they're going to move towards the infection in a process called positive chemotaxis. That's just the idea of following an increasing concentration of chemicals that may include chemicals from inflammatory cells, or it may include toxins that are produced by the bacteria. But all I have to do is follow an increasing concentration, and I get to the site of the infection. And then last, okay, so now the white blood cell got here, we've got to be able to stick to it. We've got to be able to grab it or adhere to it. So this is how the phagocyte attaches to its target. Now the phagocyte's going to have receptors on its cell membrane to be able to bind to structures on the bacteria. But sometimes what the white blood cell or the phagocyte has to grab a hold of is teeny, teeny, tiny, and it can't really grab a hold of it effectively. So it can't adhere to the bacteria. So we need help. And there's certain chemicals in our immune system that help in this process of grabbing or of the phagocyte grabbing to or adhering to the pathogen. And that's a process called opsonization. Again, think of it in this sense, that the phagocyte has hands to grab a hold of little teeny handles on the bacteria. But if that handles on the bacteria are so little, it's not very good at grabbing a hold of that bacteria so it engulf it. So what these chemicals do that allow for opsonization, they just make big handles. They turn those little teeny handles of the bacteria into big handles, and now the neutrophil can grab a hold of these big handles and then be able to engulf the bacteria and get rid of it. Okay, so again, enhancing that ability to adhere to a pathogen is what's called opsonization. Then, of course, the last step is phagocytosis. That is, the phagocyte has to engulf the bacteria. And here's phagocytosis. The idea here is a microbe that this phagocyte is going to engulf by sticking out pseudopods, little extensions of its cytoplasm. And now the bacteria is inside of the phagocyte in a vesicle. That vesicle is going to combine with a lysosome that contains digestive enzymes that digests it up, and then the indigestible material is going to be deposited out as waste products. Now, what the phagocyte uses to basically destroy this bacteria can vary. It could include things like nitric oxide, hydrogen peroxide, and defensin. Defensin actually will punch holes in the bacteria and cause the cell to lyse, and then you can digest it up that way and get rid of its parts. Or it could be lysozyme, which is typically the enzyme that's used to digest it up. Other things that macrophages and dendritic cells can do besides phagocytosis is they can act as sentinels, and this is involves what's called a toll-like receptor. These are receptors on the macrophages that can recognize different classes of microbes. So there's like 11 types of microbes out there, and they'll recognize those 11 types. And so think of these guys as sentinels. They're basically on the lookout for particular kinds of microbes, and then in response to that, they send an alert out. And that is technically it's not through obviously a little walkie-talkie here, but it's by the releasing cytokines. Again, these are chemical messengers, and those chemical messengers, they'll then trigger inflammation, which will bring in this huge front of phagocytes and other chemicals to get rid of the pathogen that they recognized. Another thing that macrophages and dendritic cells will do is what's called antigen presenting. That is, they're going to show the T helper cells of our adaptive immune system what they've found and what they've engulfed. And so think of antigen presenting cells as scouts. In any army, you're going to have scouts that have to go ahead of the troops and go scout looking for the enemy. And so these guys, this is basically what macrophages do. They're going to go out and look for the enemy and say they find the enemy, they engulf it, and then they come back here and tell what's the T helper cell. Now the T helper cell is kind of our general. And so this macrophage goes up to the T helper cell and says, hey, look what I found. Now the general is all excited about the scout reporting back to him, and then he's going to basically, through the release of a bunch of chemicals, excite other T cells and the B cells and get them to have an immune response and to fight off the pathogen. So again, antigen presenting cells are scouts. They're looking for and reporting back to the T cell and telling them what they've found. Now, mast cells are also involved in our innate defenses. Now, mast cells, I've always mentioned, are kind of related to allergies because it is the mast cells that we can blame for our hay fever, for example. But it does do a lot of other things. So here's a mast cell here. Now, this mast cell in this picture has been what's called sensitized because he's got IgE antibodies attached to him. The first time you're exposed to an antigen or an allergen, your B cells will start making antibodies, and those IgE antibodies will attach to mast cells. And so every time you're exposed to that allergen again, that binding of the allergen or the antigen to the IgE antibodies triggers the release of histamine. Sometimes you'll hear it called the degranulation. You're basically dumping all these little vesicles that contain histamine out. And so in an allergic response, or those people who have hay fever, the amount of histamine released is way too much. It's like a huge amount. And so you get a lot of histamine released, you get a lot of vasodilation, a lot of vascular permeability. That means your blood vessels in your nose, in the nasal passages, they dilate and they get leaky, and so all kinds of fluid leaks out of those capillaries, and your nose runs and you feel like crap because you now can't breathe and you have an allergy. Now, normal levels of histamine, though, also cause those things, but it's also to increase inflammation. That is to a good inflammation, because we have some allergen there. We want to make sure we get all the cells involved and make the cells leak, or make the blood vessels leaky, so we can get cells there to take care of this antigen that's invaded our body. Okay, say it be now I'm talking about foreign antigen in this case. Other things histamine can do too is increase mucus production, thereby trapping the pathogen in the mucus, so it's easier for white blood cells to get to it and engulf it. Mast cells are capable of phagocytosis, and they also can act as antigen-presenting cells, or our scouts. And they release chemicals, cytokines. These are chemotactic factors. This means that they release chemicals that dendritic cells and neutrophils and natural killer cells can follow and use positive chemotaxis, follow the trail, and so that recruits those cells or brings those cells to wherever the mast cells are. And these guys, remember, are the ones, the dendritic cells and neutrophils are going to phagocytize whatever it is that's bothering us, and natural killer cells will kill. So we'll look at those guys next. Natural killer cells, again, are just what they are named. They kill cells. They are for what's called immunological surveillance. Now, in immunological surveillance, what's happening is these natural killer cells go around and check out your body cells. And they want to know, is the body cell normal or is it abnormal? It's abnormal if it has things like a virus infected in it, it's a cancer cell, or maybe there's a bacteria infected inside of it. And so it wants to get rid of those body cells that are no good, because obviously if it's infected with a virus, that cell's more worried about producing viruses. Bacteria are hiding in there. We don't want them hiding inside the house. Cancer cells obviously are bad. And so that natural killer cell is going to destroy those cells that are no longer any good to us. And here's a picture of a natural killer cell actually attacking a cancer cell. Now they do this by identifying cells that lack what are called self markers, or think of those as dog tags. And this is how they do it. The natural killer cells use what's called an MHC Class 1. Now, MHC Class 1 are a type of marker on your body cells that say, I belong to me. So there are dog tags. Everybody has different MHC Class 1. Nobody except maybe identical twins will have the exact same MHC Class 1. Okay, so they're all unique. So that way I can recognize self from non-self. So here is a normal cell. And this natural killer cell is going to see, is this body cell normal or not? Okay. Well, if it's normal, it has on it these activation receptors that the natural killer cell can bind to, and it's got an MHC Class 1 that the natural killer cell binds to. So it's got the two different kinds of receptors. Now, the activation receptor is just that. It activates natural killer cells, and that is a positive reaction that's going to make the natural killer cell want to kill that cell. So we don't want it, because remember, this is a normal cell. We don't want that one killed. So we need to inhibit it. We need to stop the natural killer cell from killing normal cells, and we use the dog tags, the MHC Class 1s, as a way to say, hey, don't kill me, I'm good. So this binding of the MHC Class 1 with a receptor inhibits the natural killer cell from wanting to kill this cell. So since we have an activation receptor and an MHC Class 1, that MHC Class 1 inhibits or counteracts the activation one, and the natural killer cells go, oh, okay, you're normal, I'll leave you alone. But now let's say we've got a cell where it's a cancer cell down here. Now in a cancer cell, or even a cell infected with a virus or bacteria, can't make normal stuff, and it stops making MHC Class 1s. Or maybe it'll make weird ones. But in this example here, let's say it doesn't make an MHC Class 1. So now it still has the activation receptor, so that activates the natural killer cell. But now, since there's no MHC Class 1, there's nothing to inhibit that natural killer cell. So he's been activated, but no inhibition means he's going to produce his stuff, and he's going to kill that target cell. Now, once the natural killer cell determines what's going on, and that he needs to kill him, well, let's look and see how he actually kills them. Well, he recognizes cells, of course, again, that are abnormal because they don't have that MHC Class 1 to deal with or to inhibit him from releasing things. And so what he does then, in response to being excited or activated, he releases chemicals called either perforin or granzymes. Now, the perforin, its job is to punch a hole in the cell membrane of our target cell. So let's say it's a viral infected cell. It punches a hole in that cell. That's going to allow the cell to lyse, to break, because it's going to release all of its stuff. It's going to fall out. It's like a shot. It's a bullet wound. Now that bullet wound then makes all the contents of the cytoplasm leak out, and this cell's going to die from that. It also allows the granzymes. The granzymes are enzymes that enter in through that pore into the cell and triggers a process called apoptosis or apoptotic enzymes, and that's going to activate apoptosis or cell death. And all cells have this programmed cell death that we just have to have the right enzymes there to trigger it. And so the granzymes are going to trigger basically this cell suicide. So in either case, either through the perforins or through the granzymes, this cell dies and we get rid of it. Now, cytokines, as I mentioned before, cytokines are basically chemical signaling molecules. We'll see all kinds of chemical signaling molecules or cytokines. Most of them are going to be secreted by the macrophages, the dendritic cells, and then we'll see when we do adaptive immune response that T helper cells also secrete them. Examples can include interleukin-1, interferon alpha, tumor necrosis factor, lots of different types. I don't want you to worry about which kind does what things. That's well beyond where we need to go with this. Just when you see those terms, just think, it's just a chemical signal to tell another cell to do something. And so they're going to have all kinds of functionings, include intercellular communication signals, stimulating white blood cells to be produced in the marrow, to start inflammation, or actually anti-inflammatory as well. Chemotaxis, growth factors, growth inhibitors, all kinds of different possibilities. It just depends on the situation, what we want these cytokines to do. Another chemical signal of our innate defense is interferon. Now, interferon, its job is basically to defend cells from viral infections. So it's a type of cytokine. It's a chemical messenger. It can also be activated through macrophages and natural killer cells, but I want you to look at this scenario here. Let's say this cell here gets infected with a virus. So the virus injects its DNA or RNA into the cell. That gets incorporated into the cell's DNA, and this cell is going to start making viral parts. And there's nothing you can do about it. So here's new viral parts. Those viral parts are going to be released out once we have a lot of them made. And the cell can't really stop that process. He's done, okay? So in response, though, to this, what this cell is going to do is it's going to send a warning sign to its neighbor. It's going to release interferon. Here's interferon down here. So interferon then will be released by that cell, and it's basically a warning call to adjacent body cells saying, hey, I'm infected with a virus. That virus might try to get inside you, so board your doors and windows and get your shotguns out to stop that virus from infecting you. And so that's what this cell does. It's going to start making or synthesizing things to make up a bigger defense against this nasty virus. So that way we slow down the spread of the virus. Complement is another chemical that can be used in fighting off pathogens. This is a system of proteins that are circulating around, and they may help on all kinds of different things, such as helping antibodies in destroying the pathogen. Their activation can be one of three pathways here, the classical lectin or alternative pathways. Don't worry about trying to figure out which one is which and what they are, but if you see those terms, you'll know what they're referring to. The idea here is you're basically taking a bunch of proteins that have been floating around in the blood and finally activating it. That activated now complement system can do a number of things. One is it can lead to opsonization. Remember the handles, putting out big handles on bacteria so the phagocytes can grab a hold of it? Well, here's the chemical that makes the big handles, the complement. Or it can help in leading into inflammation. So it can increase histamine release. It can get blood vessels to be permeable. It can help with chemotaxis to draw more white blood cells to the infected area, that type of thing. And then last it can make is what are called membrane attack complexes. This is very similar to what perforin does. It basically, these membrane attack complexes form holes in the cell membrane of a target cell, and therefore the cell leaks out and it lyses because it loses all of its stuff. So think of the membrane attack complex is also a bullet hole in the membrane of our target cell. So that ends our first half of our innate defenses. In the next video lecture, I'll pick up from here and talk about inflammation, how to put all these pieces that I've been talking about together into an inflammatory response.